Okay, before we take the quiz, let's go through some of this. We left off on Wednesday going through the initiation. We went through translation, or went through transcription, right, and started the basics of translation. I'm sitting over here today. You guys decided to switch it up, right? Huh? Oh. Well, I'm all thrown off now. That's it. It's all your fault. So we went through the fact that amino acids are added one by one. All right, we went through the different parts of the ribosome, the E site, the P site, the A site, and what they do. Okay? Each addition of an amino acid, right, there are elongation factors that we're not going to get into. There are initiation factors that are important in orientating the ribosome in the right spot. So we're going to simplify it down to basically the ribosome, because that's where all the action happens. Okay? But there are other factors involved. Okay? So initiation involves finding where the start codon is, binding a ribosome, and getting things started along with an initiator tRNA, the methionine brought in. Okay? Now elongation is going to be, let's keep adding amino acids to the growing peptide chain. So tRNA has to bring those amino acids in, peptide bond formation, and then translocation occurs, right? And translation proceeds in along the mRNA in a 5'. So here's your process right here, your amino end. It's really a cycle. Elongation is going to occur, right? So we already have initiation. Now, through initiation, it orientates the methionine tRNA in the P site, and then the A site's open. That accepts another tRNA that's bound to an amino acid. You get formation of the peptide bond, and then translocation occurs, whereby now the used tRNA, the empty tRNA in the E site is ejected, and now it can cycle back. You will also note that this is a energy driven process. Hydrolyzation of GTP. Okay? GTP is just like ATP. It's not necessarily used for all cellular processes, but it is used for some. Okay? GTP is utilized. It's converted, hydrolyzed. So this is an energy compensating or process, right? Energy utilizing. So we end up, and we left off here. Now, that cycle of elongation will continue, whereby tRNA comes in, peptide bond formation, translocation, exit, and then another tRNA comes in. As long as the tRNA, the codon that's in the A site, codes for another tRNA. So, but once that codon that's in the A site codes for a stop codon, there's no tRNA that can come in there. Okay? So when it reaches a stop codon, no tRNA is going to come in, thereby there's not going to be any translocation. Okay? So instead of accepting a tRNA that's going to match up anti-codon-codon, it accepts what's called a release factor. Okay? Another protein comes in, there's going to be an interaction with water, and it's going to cause the polypeptide, the two subunits of the ribosome and the RNA to separate. Okay? Because the translocation can't occur, everything breaks apart. All right? So, here's the process. You're going to get one of the stop codons comes together. Okay? UAG, UAA, UGA. No tRNA can come in there with an amino acid. The release factor binds. Okay? Once the release factor binds, it stops translocation. Okay? It promotes hydrolysis. What's hydrolysis? going to break something by utilizing water, right? Water is going to be integrated. It's going to kick off the tRNA. It's going to break the tRNA from the peptide itself. Peptide is pushed out, right? The tRNA is pushed out, and then everything separating with hydrolyzation of GTP. Right? So over the process of producing a protein, many GTPs are used. Right? Think about 2 for every amino acid that's added to the chain. Okay? And then another one to just stop it from happening. So this is an energy intensive process. Building proteins is anabolic, and it's going to build, and it's going to utilize energy. So after the release factor, right, after hydrolysis, the ribosomes separate into subunits and go off and bind another mRNA. That mRNA could be rebound to another ribosome, or it could, right at this point, already have a ribosome bound to it, right, further upstream from this ribosome. Okay, all right, or it could be degraded depending upon the needs of the cell. On the termination. So initiation, finding the right start site, orientating and binding the ribosome so that the P site is bound by a methionine tRNA.
Are we not supposed to memorize the stop codons?
You don't have to memorize the stop codons. So if I said you found a stop codon, I'm not going to say you find it runs into UGA. You should know that one. No, I'm kidding. It runs into UGA, and you don't have to know that that's a stop codon. All right. So now we have a complete polypeptide. Is that necessarily a functional protein? No, why? We have the whole we have this whole chain we have all the amino acids in the protein. Why is that not necessarily a functional protein?
It needs to be shaped.
And now it needs structure. Okay? It needs structure. It will assume its structure, and beyond that, it also may have what we call post-translational modifications. Okay? So the cell can add parts to amino acids. It can put tags on it, on the protein, to determine whether or not it's going to be functional. It can change its biologic activity based upon those modifications. Okay? So, a lot of times translation itself is just not enough to make a functional protein. It has to assume shape, and there's other modifications. All right? And it also can be target to specific sites. All right? Part of the amino acid chain itself will tell you where that protein is supposed to go in the cell. Okay? So, protein folding. Right? So during synthesis, the polypeptide chain will begin to fold. All right? Depending upon the type of protein it is. All right? There are other proteins within the cell that will direct how the fold occurs. All right? Because it's not all proteins fold into their correct shape spontaneously or on their own. Right? And a lot of times there is a directed process that has to be. Parts of the protein have to be folded before another part, right? Tab A has to go into slot B before you can change over to globular structure C, okay? And then there's also post. All right? Targeting. Proteins are different places, okay? So a protein that's going to the plasma membrane, that's going to function at the plasma membrane, has to go there, right? As opposed to if it goes to the nuclear membrane, where it doesn't function, okay? And we can the cell knows this, and we can change this, in the lab to see what the function of proteins are, all right? Ribosomes exist in 2 areas, either in the ER, attached to the endoplasmic reticulum, the rough ER, or free-floating in the cytoplasm. Okay? The endoplasmic reticulum structured how? What is it part of in terms of the parts of the cell? The endomembrane system, which is also an extension of the plasma membrane, right? So if I wanted to make, as a cell, if a cell needed to make a protein that was going to be embedded, a transmembrane integral protein in the membrane, would it make it free-floating at a ribosome in the cytosol, or would they make it already part of the membrane in the endoplasmic reticulum? What do you think? In the membrane, in the ER, or in the cytoplasm? In the ER. And that's exactly what happens. So ribosomes embedded in the ER, it can produce a protein and then integrate it directly into a chunk of the membrane, which then can come, and that whole protein, along with parts of the membrane, can go and be shuttled to the plasma membrane where it's actually going to function. Okay? That's an example of specifying location. Cytoplasmic proteins are going to be produced. All right, any proteins are going to be secreted out of the cell. All right, it's going to produce it at the ER. It's going to be produced into a vesicle, which then will be shuttled out of the cell. Right, but other proteins that are going to exist in the cytoplasm, such as the cytoskeleton, that will be a cytosolic ribosome. All right. So, synthesis always begins in the cytosol. It finishes in the cytosol unless the polypeptide signals to the ribosome to attach. So really, we're going to start at one of those free cytoplasmic, cytosolic ribosomes. That's where everything's going to start. Why does it start in the cytosol and not in the ER? Where does the mRNA go when it exits the nucleus? It goes to the cytoplasm. Goes to the cytosol, so then it's going to bind the ribosome. All right? Then from there, there's a signal peptide. There is a, a portion of the amino acid chain of the polypeptide that tells the ribosome this protein is going to be going here or going there. When that occurs, then the ribosome goes to the ER for secretion, and then it attaches and so on. Right? So there's a. How does that signal peptide work? So a signal peptide is part of, here's your ribosome, right? Here's your mRNA. Here's your growing polypeptide chain. See, this portion right there is your signal peptide. This particle, this signal recognition particle, SRP, binds to that signal peptide. All right, so it's going to bind to the signal peptide right here. Okay? And then now that SRP is going to translocate this whole complex to the ER. And then the ribosome will bind the ER. Right? And then we'll produce that protein in the endoplasmic reticulum. Okay? So it's the signal peptide, right? Or what we call a signal sequence is a sequence of amino acids which is part of the peptide chain, and that determines where that protein is going to go. It's not part of the mRNA. Okay, so this is a better version. There's your ribosome bound to the mRNA. You get the signal peptide. The SRP binds and then tethers that to a receptor on the ER. Then what that does, it links the ribosome to the ER, and now that signal, that peptide is produced into a lumen of a vesicle, right, which then can be secreted from the cell. Okay, so directing it is a function of the amino acid sequence. Questions on that? Questions on directing where proteins go and how they develop their function? Another aspect of translation is multiple copies, all right, or lots of proteins produced at once. Because it's not just we're producing one protein per mRNA, we're producing lots and lots of protein per mRNA sequence. All right, so most of the time you will end up with an mRNA that is bound by several ribosomes at once, making it, at one time several copies of that protein at once, right? Just staggered, right, at different points. What those are called are strings of ribosomes called polyribosomes, so it kind of looks like this. Here's a ribosome, another ribosome. Right, here's your mRNA, and we'll call this your growing polypeptide. Right, the direction's this way, 5' to 3'. Okay, that is a polyribosome. See what it looks like up top there. Okay, so you have multiple ribosomes bound to one mRNA. What does that equal when you're done? In this case, there's going to be, when finished, how many copies are there? One per ribosome, so you're looking at, exponentially more protein that's produced from one message, right? Then on top of that, you produce multiple messages. Now you have exponentially more protein produced from one gene. So you have bacteria and eukaryotes can also transcribe multiple RNAs from the same gene. Multiple RNAs from the same gene. So saying transcription's occurring, you're having one copy of the mRNA produced, and then before that's done, another one is also produced. Right? In bacteria, we know that there's no boundary to separate where the nucleus is and transcription is going to occur, or where the DNA is and the transcription is going to occur, from the cytoplasm where the ribosomes are. So as soon as a mRNA is being transcribed, translation can actually begin before that mRNA is even finished. So transcription and translation in prokaryotes is simultaneous, right? So it actually is ongoing. In eukaryotes, we know there's a separation. Because of that separation, transcription has to finish, editing of the RNA has to occur prior to exiting the nucleus, and then translation begins. Okay, so here's your polyribosome in bacteria. Okay, so in bacteria, you're getting production of an mRNA. So here's RNA polymerase going along, production of the mRNA. So before it's even finished, right, you're just going to have 1, 2, 3, 4, 5, 6, 7, 8, 9 copies of the protein once it is finished. Okay, so it happens simultaneously. Very quick turnover, production, quick production of a protein.
So when you have multiple RNA polymerases making multiple mRNAs,
Mhm.
you have like the topoisomerase?
You don't need topoisomerase because it's just going to it'll unwind and cause a bubble where that gene is. So it doesn't have to, nothing is completely unwound. All right, so there'll be tension upstream and tension downstream, but nothing's completely unwound. It'll just come open, and then we'll go back. All right, and as the polymerase goes through, it will resume the DNA will resume its double helix structure. Okay, so here's your overall process of transcription and translation. Okay, so we have transcription, right? Production of a primary transcript that is not necessarily ready for translation. You have RNA editing or processing that has to occur. That includes alternative splicing. So splicing, adding of the 5-methyl-cat, poly-A tail. That allows time for, allows the mRNA to leave the nucleus. It leaves the nucleus, will bind a ribosome in the cytoplasm, which then may or may not be translocated to the endoplasmic reticulum. Initiation, elongation, termination of translation, you get production of a polypeptide. That polypeptide has to assume shape and can be post-translationally modified in order to produce a functional protein. Okay? That's the overall process. Questions on the process. On Wednesday, we'll go through some mutations, some problems that happen in the process. All right? So, we will finish chapter 14 on Wednesday. Okay? And your exam is on Friday. So we'll cover chapter 13 and chapter 14. Okay, Sienna. Yes. That's in gray too. I gotta switch it over to blue now. Okay. Other questions?